dynamics, and of course, one of the pioneers together with Klaus and some other colleagues in the problem of seizure anticipation. And today, you will talk us about a little bit about this and your work. Please, Hitten.
Great. Thank you. I hope you can all hear me. Thank you very much for the invitation. This is my first time in Brazil. I've been here for 3 hours, I think. I just landed this morning. So I'm an electrical, computer, and biomedical engineer by training, an associate professor in neurology at Yale, and the co-director of a research group that we call the Yale Neuroscience Neuroanalytics, or YNN. My fellow co-directors are Dr. Dennis Spencer, a neurosurgeon, Tor Eid, who does lab medicine work. We are a very clinical translational group. We're right at the edge of bringing new treatments to bear on epilepsy. Our work is threefold, primarily research on epilepsy, where we try to understand how the condition of epilepsy or epileptogenesis arises, how seizures arise, ictogenesis, the stage, the transition, when you go from ongoing activity like I'm having right now, and all of a sudden my attention could move here for 10 seconds, and I would come back and talk to you. And in those 10 seconds, my brain has transitioned into another state where its normal function has been interrupted. And that's what we try to understand. How does this happen to an otherwise normal person who's going through these state transitions and then back into normal behavior? We are trying to understand where the seizures start from, localizing the seizure onset area in the brain. We try to forecast seizures before they happen, because if you can do that, then we could stop them. We believe strongly in the network theory of epilepsy, and this is some work that we do in order to try to prove this or better establish it. We study patients with intractable epilepsy who are undergoing epilepsy, surgery for epilepsy. We also study animal models of epilepsy. Separately, in the second area, we develop neurotechnology to monitor and modulate the brain. Here, we are developing sensors, electronics, brain monitoring systems, brain-computer interfaces. And the third part is we train people. We mentor in our lab undergraduate students, graduate students, medical students, postdoctoral fellows, and junior faculty. We have more than 25 such individuals at different stages of their training. I'm going to be talking about 4 of my projects. These are 4 disparate projects, but you'll get a sense of what we do in the lab through this work. But to start with, the focal theory of epilepsy holds that seizures arise from a focal area of the brain, and that focal area recruits surrounding normal cortex in this aberrant behavior, and through that, builds it and takes it into a seizure, and at some point that seizure ends. We don't understand these transitions well, and that's part of the reason I'm here. This implicates a sort of, nominally the balance between excitation, inhibition. The story is that essentially, excitation overruns, overcomes inhibition surrounded, and then it recruits normal tissue in this activity. And so it's an out-of-control excitation leading to seizure. An alternate sort of statement on seizures has been sort of postulated 20 years back, a little more than 20 years back, by my postdoc mentor, Dr. Susan Spencer, which essentially says that seizures arise from an aberrant brain network, a large-scale brain network. I've shown this here only as 2 nodes and 2 edges connecting the 2 nodes. I'm not saying anything more about that at this point. But the 4 projects that that we work on are all sort of in one way or the other influenced by the network theory of epilepsy. The 4 projects are what we call the NeuroProbe project, the Yale Brain Atlas, seizure forecasting, and Yale sort of network brain-computer interface. We'll go through each one at a time. The first project is funded by NIH to Dr. Spencer and myself. It's a large grant in which we must take technology from sensors through its development, through the FDA, get permission from the FDA, and then do a study in 10 patients at Yale. We're making a brain implantable device. It looks like that. What it does, well, why are we doing this? This is for use in the neuro ICU. We have patients that come in. One thing about brain injury that's separate from injury to any other part of your body, if you break a limb and you go into the hospital, you will recover. The trauma, whatever trauma you've faced, the minute you step into the hospital, a recovery starts. This is not the case with brain injury. With brain injury, you could go into the hospital, you could be in the best ICU, but in roughly 40% of the subjects or patients, a secondary brain injury starts, which unless we are monitoring that from within the brain, we miss the signs for it. So we need sensors that are placed inside the brain to pick up hypoxia, ischemia, et cetera. And if you don't pick it up, then this leads to secondary brain injury, death, sort of neuronal death and death. The way this is monitored currently, it entails placing multiple monitors into the brain directly. And each probe sort of requires separate wiring, calibration, maintenance, training. There's a lack of synchronization. All this data goes to different monitors. Here I have a photograph of, you can see how complicated this is behind the patient's bed. It's a mess of wiring, monitors, electronics. Each one of them is a separate system. The data doesn't even go to a single system on a single clock so that we can analyze it together. So you've got pressure going to one monitor, temperature going to another monitor, oxygen going to another monitor, EEG going to another monitor. We have to invest in a data aggregator to bring all the data together. So what we are doing, and also these are different probes placed targeting different parts of the brain, require larger opening in the skull. So what we're doing is a very simple thing, engineering. We're combining all the modalities into a single probe, and then we call that a single multimodal brain probe, single set of electronics, a single monitor. We're building all of those things in the lab with equal or better sensitivity, single point synchronized sensor data, single output connection. We've also changed this so that no surgeon's needed for the placement. This can be done without an OR, and it's portable overall. The goal ultimately is to collect the data, stream it to the cloud for processing and analysis, so you get this nice, strongly multimodal data stream synchronized going into the cloud. The way this is being targeted in this grant is, we're making the system that's shown here, sensors, electronics, monitor, interfacing with electronics and other monitors. And in a two-phase, we're required by NIH to do this in 2 phases. The first phase is to build all the equipment, take it through the FDA. The second phase is to do the early feasibility study in humans. There are many competitive advantages from having a single device that does everything, and this just outlines all of them. It's a single probe, single access site into the brain, and the costs, et cetera, are improved as well. In progress to date, we've developed the sensors in the lab in our clean rooms, developed patent portfolio, completed 2 FDA pre-submissions, developed prototype devices, matched the performance of predicate devices, initiated all the things that are needed to transfer this into commercialization, design for manufacturing, set up our own quality management system, design control, et cetera. We are in the process of going to the FDA for regulatory approval, which will probably happen in the next year or in the next 12 to 14 months. And then we'll be conducting early feasibility study at Yale. And this just shows the probe, a 3D model of the probe, and being able to get all the data on a single screen. Also in the lab, we've got postdocs and junior faculty, Dr. Gu from China, Dr. Deshane Murray from the UK, Jesus from Mexico. They work on building new sensors. So this shows wafer with oxygen sensors on it that we built and designed ourselves. This shows the construction of fluidics chamber and a chip that has got sensors for measuring neurochemistry. We would like to bring in these new modalities into the brain. We build the neurochemistry sensors on the back of a transistor, and we have what we call silicon nanowires that can be functionalized to detect different neurochemistry. Where we're going with this is to try and get as much a rich representation of the dynamical changes within the brain as possible, because we believe we're missing certain key information. The team's quite large. I don't have everyone's photograph here. We've got faculty in mathematics who work with us, and this is the clinical team that are waiting for us to complete the work here and then take it into the human study. A second project is more computational in nature, and here the challenge we are facing is that the brain is quite difficult to communicate and standardize over. The locations, if you're over just a slight distance away from another location, we don't know how to standardize our communication about that. So what we did, and we also noticed that a lot of groups have built brain atlases. The brain atlases typically are built using computational methods from fMRI data. For example, you do correlations, you use network measures in order to then define subparts of the brain that are operating similarly. We did not want to do that because then that sets up a circular sort of reasoning where you have a network-defined atlas being used to study networks. So we built the highest resolution brain atlas, at least to our knowledge, it's the highest resolution anatomical brain atlas, where we broke the cortex into 690 parts. This was done a lot by Harry McGrath, who was at King's College London at that time, worked with us for over a year, and this is a decomposition of the brain into 690 parcels, and including the insula, hippocampus, amygdala, all the gray matter on the brain. And then we've got other students. This is an interesting project in that we only have students working on it, and only undergraduate students. So Alex King, who's graduated from UC Berkeley now, has sort of measured cortical thickness on this atlas. Omar Chishti, who's doing his graduate studies at Max Planck in Berlin, worked out white matter connectivity between every pair of 690 parcels. So every point to every point, we've got white matter streamlines and connectivity and distance. Elizabeth Watson, who's a senior, will be graduating in May, has taken all this information plus information that we have on language mapping from the literature, from other databases, and brought that all into the brain atlas, including electrical stimulation data, so that we can co-locate in this standardized space language and brain function information. Evan, who's currently doing his PhD at MIT, when he was undergrad with us, took a lot of this data. He took structural data from MRI information, the white matter connectivity, which Omar had worked up, took this into a number of structural measurements, 13 different structural measurements, and also functional measurements that have been tied from large repositories of fMRI data. And so he brought this computed measures of structure and function together to try to understand certain key questions. Is the structure-function relationship the same throughout the brain? Does it depend on the function being studied? And essentially what he showed is that it exists on a gradient. It's stronger. Structure and function are strongly tied together in primary sensory areas, motor areas, areas of the brain where the function is cleanly delineated and sort of is a single function, if you wish. But as you go into association cortex, the coupling between structure and function becomes weaker. And what we've postulated based on that is that one possible reason is that the evolution of the human brain may explain this gradient, is that while direct connections between different regions were sufficient for faculties such as vision and movement, as the brain developed more advanced capabilities like complex cognition, these direct connections had maxed out the usefulness. Hence, the brain developed more indirect connections between regions to establish more advanced capabilities. And so these are what we see when we move out of the primary areas of the brain. And this was published by Evan in Nature Communications. This data is all sitting on our website. You can find it. And we're writing up, he's written up a methods paper, which we are hoping to publish soon. It should be submitted in the next week or so. Sorry, I should show that. It's, as I mentioned, a lot of students. These are all undergrad students, and then this is Ming. Sorry, one of them, Yi, is a graduate student. Ming and the rest of us are the faculty who work with these students. So this is our second project, purely computational in nature, in order to standardize brain anatomy. And we're also bringing in function, studies of function into this. A third project, which started last year, is not as well developed yet. Here we are trying to work on understanding certain signals that we see from patients who have epilepsy. The trace on top shows 100+ days of recording made by a brain implantable device in one of our patients. This is the NeuroPace responsive neurostimulator device. It records from a few locations, and when we look at the detections, we see this cyclical change, and we see these cyclical sort of manifestations in both men and women. They can be cycles 7 days, 14 days, 30 days. We don't understand what these are, but seizures tend to arise from the higher points of these cyclical changes, which I've marked in red, and seizures are seldom seen in the lower points. So what we are doing here is we're collecting saliva samples from these individuals as they go about their daily life, and then we send that saliva through a mass spec analysis method, break it down into its constituents, and then we're sending that through machine learning methods to try and understand if we can detect those cycles that are emerging from the brain in the saliva for the person. Then we could tell the person whether they're going to have a good day or a bad day. It's in this way that we do take our work into sort of clinical translation. We're also just trying to understand the relationship between neurochemistry and the body's chemistry and the saliva chemistry. A fourth project, and this will be my last project, is where we are building a brain-computer interface, again, influenced by our need for studying brain, monitoring and modulating brain networks. Now, in the previous slide, I showed you a brain-computer interface device for epilepsy. It's revolutionary in its own right, the NeuroPace device. It's roughly 3,000 human beings in the US that have this device to help them control their seizures. It monitors from very few locations, and the monitoring that it does is very limited. It has a few feature detectors running. When you meet the criteria, you cross a threshold on the feature detector, it stimulates. But this device is not meant to monitor a brain network or modulate a brain network. So it's revolutionary in the sense that you have closed-loop feedback control in patients who are walking around, living their daily life. But at the same time, there's a need, if we think this is a network disorder, and we need to monitor a network and modulate a network, there's a need for us to enhance its capabilities. So what we are doing here, and this is with colleagues, Dr. Manohar and Bhattacharjee at Yale in electrical engineering, computer science, we're building our own chips. And the question is, can we build a brain-computer interface that can support a large channel count and high data rate? So you translated this problem into an engineering problem. Support state-of-the-art computational techniques, support machine learning, deep learning, connectivity analysis. And I'm listening to the conversation here and hoping to understand what methods might be emerging, because we are at the stage of designing these chips. They take 18 to 24 months to design and fabricate and bring to bear into the lab. So we're always on the lookout for what should be programmed. We would like the device to be software programmable, so the same hardware can be reused based on new knowledge and new clinical uses, and last for years on a single battery. We've generated our first chip. This is not vaporware. We've got the actual chips now. It's got a power budget of 15 milliwatts for its computation, and it sort of supports, it's programmable, it supports up to 96 channels, can sort of sample 96 channels, stimulate 16 channels, it has a sampling frequency up to 30 kilohertz, up to 16-bit A to D resolution, and it's, I'm supposed to be getting the first of these chips interfaced with our animal monitoring system in this quarter or the next quarter, so we're quite excited to see what we can do with it. And then we're preparing to make the next 2 generations of chips. We've got funding for 2 more generations of chips. The way we do this is we're building processing elements, a bit like how the chips are designed for your cell phone. There are different clock domains within your cell phone, and processing elements are hardware coded. So this is compression algorithm, discrete wavelet transform, energy operator, Fourier transform, cross-correlation, bandpass filtering, support vector machines. All of these are different hardware that's been sort of created and put together in this as a single element, single chip and it's got a RISC-V sort of microcontroller as well. And that's how we sort of approach this problem of decreasing the power consumption, and that's the team that's been working on it. So those are the 4 projects that I wanted to present to you. That's all work that's going on in our research group. And as I mentioned, we are quite strongly influenced by the network theory of epilepsy. We are trying to understand what aspects of the brain need to be monitored and how we can modulate a brain network to keep a person from going into a seizure and or at least minimize the time spent in a seizure. Thanks a lot for your attention. Thank you for the invitation.
No, I have some questions, Hitten, especially about your second project, because it's a very, as a neuroscientist working on brain networks, I often realize that some colleagues or students mainly, that they have a confusion, and the number of the areas plays an important role on the definition of the network. And according to your experience, what is your take on that? Because, for instance, here for your atlas, is there any particular advantage over other atlas, or because it's very, the parcellation is relatively fine here?
Yeah.
Extremely fine compared, for instance, with the Lausanne atlas. And of course, the network here, I don't know how many regions.
690
Yes,
parcels.
it's quite interesting. But what is your take on that? I suppose that there are some problem for estimating the fibers.
So when we were designing this, we tried to understand what should motivate the spatial resolution. And the argument I was making is that it should respect the information that we're generating with clinical electrodes, which have a 10 millimeter center-to-center spacing. So the size of each parcel is down to 1 square centimeter of cortical surface. And this can be further broken down if you want to go even finer. You can take 1 square centimeter and break it down. But we think this is fine enough a resolution for the information we're collecting with a number of modalities. So as shown by the students, they've taken electrical activity, stimulation measurements, fMRI, MRI, CT, PET, different modalities onto the space. Some of them have much poorer resolution, and some have equal sort of resolution, if you wish, and have found useful information at this spatial resolution. Now, to take this to the point of networks, so the other student that worked on this was Omar, where he did this white matter connectivity between every pair of parcels. And we found that the white matter connectivity is quite sparse, actually, when you work it out. Now, there are many issues with working out white matter tractograms, and many things need to be resolved, but this is the final connectivity matrix at this 690 by 690 space is quite sparse. So we're hoping that we can build up from that and look at regions, gyri, et cetera, or larger areas if you've got the finer description. It's easier for us to build up. And so we didn't want to limit it in that fashion. But we didn't know about the white matter connectivity when we started. We used the argument of a 1 square centimeter spatial resolution as our starting point.
Just a very elementary question. I didn't get exactly how the neuroprobe works. I mean, how do you measure, I mean, the EEG? And you seem to have several, so then I didn't understand.
Sure. Yeah. So the neuroprobe has got a sensor shelf at the tip, which holds sensors for pressure, temperature, and oxygen. We build our own, so pressure, they all fit in here. And then these are contacts for electrical activity. This is essentially bare metal that's in touch with the tissue. And we pick up the difference between these contacts, 2, 3, and 4 versus this. This serves as the reference contact. It's in white matter, which is relatively neutral, if you wish, electrically, while the other contacts are sitting in gray matter where the activity is. So there's a tiny potential that's picked up as the difference between the two, and that gets amplified. So that's essentially, it's the potential difference is what's being between 2, 3, and 4, and 1 that we are measuring. And then the other sensors are all in the shelf here.
Hitten, sorry. Question about this, it's very interesting, dispositive of the neuroprobe, and it's supposed to be implanted for how many time for this patient? Because this is not implanted in epileptic patients, this is implanted with injured patients.
So
how many times do they have?
When we proposed, we are an epilepsy group. When we proposed it, we wanted it for epilepsy patients. One of the reasons we wanted this is we have networks which are being defined based on electrical measurements, from EEG, and networks being defined from fMRI measurements based on blood oxygenation. We wanted a device that could measure both oxygen and EEG from the same location in order to try to bring these different pieces of information closer together. The funding agency said we were too ambitious and we also wanted to measure neurochemistry. They said we were too ambitious and too aggressive and told us to step back and focus and remove the neurochemistry and focus just on pressure, temperature, oxygen, EEG, and target traumatic brain injury. And they were very correct, actually, because that simplified our path through the FDA. Otherwise, the path through the FDA would have been much more complicated. For this, there are predicate devices for each of these modalities, so our path is much simpler. And so that's why we built it in this fashion. But our hope is that this would be taken into epilepsy patients in the future. For traumatic brain injury, typically 7 days is the duration. Okay. For epilepsy patients, it might be slightly longer during intracranial EEG monitoring. We're designing it for up to 28 days.
Wow, great. I mean, if it works for, I mean, if it's approved, it would be great, especially for the oxygen and the neuro. Yeah. Is there any other question? No, Marcus?
It's designed to only go into, typically in traumatic brain injury, they go into white matter. They don't go very deep in. So it's just a few centimeters. It goes into the frontal lobe, through the skull, through the gray matter, and the tip is in white matter.
Great. Thank you. I think we can thank Hitten for his lecture. Thank you, Hitten.
Thank you very much.
So now we have